Hello, everyone, and welcome back. It's been a while since we've had a lecture here. I kind of wanted to take some time off to let you all decompress and process the stuff that we've gone through with both our lab practical and the major tests that we had last week for the nervous system and for the muscular system. That's probably the toughest portion of A&P. So for this last couple of lecture notes, and if you look on Blackboard, you'll notice that we only have 2 sets of lecture notes that are left for us. We have the endocrine notes, and then we also have the brain, spinal cord, senses notes, or brain reflexes, autonomic nervous system. So there are really only 2 units of notes that are left. We'll finish the endocrine notes this week, and we'll get started on the brain reflexes and the autonomic nervous system next week. And we might get a chance to get started on the last set of notes now before Thanksgiving break, but the only day that we'll have a lecture will be on the 23rd, because we won't have one that Thursday because that's going to be Thanksgiving Day. So if you look at your calendars, you'll notice that we're really kind of winding down here. This week we will finish the endocrine notes, and then the next week we will start, maybe have an opportunity to finish to see how far we get through today, the last set of notes. And then the last, first week in December, last week in November, we're going to have our final lecture exam. So your final lecture exam is going to be the week of November 30th through December 4th. So that week there, and we'll kind of do the same thing on that Wednesday. I'll have it Wednesday evening on December 2nd. You can take that exam, or you can also take that exam on Thursday during the day sometime. I think it's from like 9 until 1, you can take that exam. So we'll have that exam happen, our last lecture exam, and it's only going to cover 2 chapters again, or 2 units, the endocrine system and the brain, spinal cord, and autonomic nervous system. I will post all of this stuff in an announcement. Then the very next week, the week of December 7th, you'll have your final lab practical. And the final lab practical will be the sheep brain dissection, the cow eye, the special senses of the ear, and all those cranial nerves. So that will be your very last lab practical. And then from there, we have our final exam. Remember, your final exam is going to be comprehensive, so everything we've done from August until the end of our autonomic nervous system notes will be on your final, but it is optional. If you're happy with your exam grades up to the final, you don't have to take it. You can keep that as your final grade. If you would like to replace one of your exam grades with your final grade, you can do that, but I only replace it a grade if it's higher, so the final exam can't hurt you at all. It can only help you. So just a couple of housekeeping things, because it's been a while since we've really talked about what's going on. So remember, logistically, what's going to happen this week, we'll finish endocrine and perhaps start our last set of notes. Next week, we'll start our last set of notes, perhaps finish it, and then our exam is going to be the week of November 30th through December 4th for our lecture exam, and then you'll have December 7th will be your last lab practical. It's going to happen there. All right, so if you have any questions, comments, or concerns, please let me know via email, and I'm going to take all these little things off. Or if you're hanging out in here, any questions, comments, or concerns that you may have. Great question, Tanya. No, we are not scheduled for a quiz today. We're scheduled for a quiz, but we're not having a quiz today because we don't have anything to have the quiz over. So no, we're not having this quiz today. We won't be having that. Good question. I need to make sure I put that on the announcement. Thank you for reminding me of that. All right, so the endocrine system. The endocrine system is in many ways is going to parallel your nervous system, considering that it's able to send and receive messages in much the same way that neurotransmitters can send messages on opening those channels or telling which channels to open and how the cell is going to respond to it. Big difference between the endocrine system and the nervous system is timing. The endocrine system is going to regulate what we consider to be long-term process. So growth is totally something that takes a while. Development, reproduction, all of those are things that can't just happen overnight. With the nervous system, those synaptic transmissions and those messages are going to be sent fairly quickly, whereas with the endocrine system, it's more of a long-term process. So that's one very big difference between the two. Like the nervous system, the endocrine system also has special chemical units that are meant to be messengers, except that these chemical messengers, instead of being called neurotransmitters as we would have them in the nervous system, they're called hormones. And they relay information and instructions between cells, and how those cells respond to those instructions it's based on the target cell itself. So in much the same way that the neurotransmitters didn't really tell the cell how to respond, they just sent the message. Hormones do the same thing. They just send the message. How that cell responds to that message is completely determined by the target cell. So endocrine communication, endocrine cells are going to release hormones, and those are those chemical messengers into the bloodstream. And what these hormones can do is that when they bind to the receptors of the cell, they can alter metabolic activities of different tissues and different organs all at the exact same time. So even though these organs or tissues, because it's making its way to these target areas via the bloodstream, they don't have to all be part of the same tissue or the same organ system. And they're definitely not going to respond all the same way. But they're able to, the hormones, when they're released and received by the target cells, that they have a widespread effect. I guess that's the term that we're looking for here. So target cells are the specific cells that process they have the receptors and they process the information. So they're going to be the ones that read the hormonal messages. If a cell is missing the specific receptors, it can't read the message from the hormone. Hormones themselves can synthesize or build different enzymes and structural proteins. They can tell the cell, okay, you need to make these certain types of enzymes, you need to make these certain types of structural proteins, you need to behave in this certain type of way. That's what the message is being sent via the hormones to the target cells. So the hormones are sending the message on how this target cell should now behave in the presence of this hormone. Hormones can also increase or decrease the rate of synthesis, or they can turn on existing enzymes or membrane channels. They can turn them on or they can turn them off. So hormones there are a variety of different things that they can cause the target cell to do. And that's different for every target cell. So the hormones that we're going to talk about can be divided into three groups. They can be amino acid derivatives, peptide hormones, or lipid derivatives. And depending on the type of hormone it is, it can alter how that hormone affects the target cell, whether it binds to a receptor or whether the hormone goes internally into the cell and it binds to the DNA and causes genes to be turned on or turned off. So the type of grouping the hormone fits into kind of has a role to play in how that hormone affects the target cells. We have some hormones that just kind of circulate freely around, and some that are bound to transport proteins. So the first group we'll talk about are those free hormones. Free hormones tend to be functional for a shorter amount of time than hormones that are bound to transport proteins. So free hormones, those that are just kind of circulating and floating around, they're not actually bound to any protein, they remain functional for less than an hour. So they're going to very quickly diffuse out of the bloodstream because that's the way hormones are sent, through the circulatory system. They bind to the target and the receptor cells, then they're quickly broken down and absorbed by either the liver or the kidney, or they can be broken down by certain enzymes in your plasma or your interstitial fluid. Big take-home message here is that these free hormones are not going to remain in circulation for as long a period of time as the hormones that are bound to proteins. Hormones that are bound to proteins are going to stay in circulation much longer because they are bound to these proteins. They're not as easily broken down and recycled by your liver and your kidneys. So they're going to remain in your bloodstream for a much longer time. And most of these, a great example of hormones that are bound to transport proteins are thyroid and steroid hormones. And if you think about what thyroid and steroid hormones do, thyroid hormones have a big role to play on metabolism, and steroid hormones have a huge role to play in reproductive development. You think about estrogen and testosterone, that the process of going through puberty, if you will, because that's probably what you're thinking of when you think of estrogen and testosterone, is it's a much longer process. You don't just wake up one morning and you have body hair in places that you didn't have it. It may seem like that, but the process of puberty takes a few years to go through. So these hormones are going to stay in circulation a much longer period in time. And as a result of them staying in circulation for a longer period in time, they're able to allow the process of growth and development to take place in incremental units, as opposed to just snapping your finger and it happening all of a sudden. More than 99% of thyroid and steroid hormones are attached to these transport proteins. So a hormone receptor is a special molecule that's on the surface of a cell, and it's where the hormone can bind to. Keep in mind, we had those 3 different classes of hormones, that not all of them are going to bind to the receptors on the surface of the cell. Some of them can bind to receptors in the nucleus or on the DNA. Hormone receptors can respond to several different hormones, so it's not that one receptor is unique just for one hormone. There are many hormones that can possibly bind to this receptor, but they may cause the cell to act differently. For example, estrogen and testosterone are 2 different hormones, but they can bind to the same receptor, and depending on whether it's estrogen that binds or testosterone that binds, it can cause that cell to act accordingly. It can cause it to act differently. Different tissues have different combinations of receptors, and different proteins or the different enzymes that they'll synthesize. And important to note that the presence or absence of a specific receptor determines how sensitive the target cell is to that hormone. So if it doesn't have the receptors for a specific hormone, it may become more or less sensitive to it. Hormones and plasma membrane receptors, because remember, as I said before, not all hormones have to bind to the plasma membrane, so some of them can actually go into the nucleus and bind to the DNA and turn genes on or turn genes off. Catecholamines and peptide hormones are 2 examples of the classes of hormones that will bind to membrane receptors. These types of hormones can't get into the cell. They're not lipid soluble. So since they can't get into the cell, they really don't have an ability to bind intracellularly. They have to bind to the plasma membrane. And that's because they're not lipid soluble. Remember, the plasma membrane is made out of a phospholipid bilayer. They bind to receptor proteins on the outer surface of the plasma membrane, and these would obviously be extracellular receptors because they can't get through the plasma membrane. They don't have these hormones that bind to the plasma membrane receptors cannot have a direct effect on activities inside the cell. They go through a second messenger system. So they use the intracellular intermediary to exert its effects. And we've talked about second messenger systems for just a little bit at the end of the nervous system. We talked about how the first messenger was the neurotransmitter, and then it was coupled with a G protein to lead to the second messenger, which could be cyclic AMP, and then cyclic AMP or would then catalyze the opening of these different channels. So we've kind of talked about very briefly at the very end of our nervous system notes second messenger systems and how they work. So some hormones can also direct the activities of the cell through the second messenger system. In this case, instead of the neurotransmitter being the first messenger, it's the hormone that's your first messenger. That first messenger then leads to the second messenger coupling with a G protein, and that second messenger a lot of times is something like cyclic AMP. That second messenger will then direct the activities in the cell. So it can result in changes in the metabolic reactions, so what's getting made. And remember, metabolism is not just your body's ability to break things down, but it's also your body's ability to build things up. So things being built up, things being broken down, the manufacture of different kinds of lipids or proteins or enzymes or whatever activities we want the cell to engage in. The second messenger, usually cyclic AMP, sometimes it's other things, we're just going to use cyclic AMP for now, will tell the cell what to do. But remember, the second messenger can't give its message until we have the binding of the hormone or the first messenger. So some important second messengers, one we talked about already, cyclic AMP. Cyclic GMP is a second messenger, and now we have yet another role of calcium. So we talked about how calcium is responsible for helping your bones maintain their strength. So we have that calcium phosphate that helps the bones to stay nice and strong. We talked about the role of calcium in muscle contractions, that it binds to troponin and it causes tropomyosin to roll over the active site and allows for the myosin head to attach to it, and you get a shortening of the sarcomere for a contraction to take place. We even talked about calcium in the nervous system. So in the nervous system, calcium had a role of releasing the neurotransmitter acetylcholine. So when calcium came into that presynaptic cell, it triggered the release of acetylcholine. Now we have another role of calcium in the endocrine system. And we'll even, in the first part of AMP2, talk about yet another role of calcium. So it's a pretty important ion. So the calcium ions here, they can also act as second messengers in the endocrine system. So this process of amplification, when we talk about the mechanisms or the how hormone action takes place as we continue this conversation, is that the binding of a small number of hormones to a molecule or membrane receptors can lead to many different second messengers in the cell. So right now we're not talking about those direct effects or any of those hormones that can just cross the plasma membrane and, like thyroid hormones, can just bind to different parts inside the cell or the nucleus or the DNA. We're talking about those hormones that have to bind to the plasma membrane that work through the second messenger system. So the process of amplification entails just maybe a small number of hormones binding to those receptors, but that cascade of events leading to the second messenger will make many, many, many of these second messenger molecules, whether it's cyclic AMP, cyclic GMP, or calcium, depending on the cell, the target cell that kind of determines what the second messenger will be. You'll have lots of these second messengers that are released so that the activities, the metabolic activities that the second messenger dictates to happen in the cell are going to be increased. So it really is this process of amplification. It's making this message louder. It's making this this response larger than it would normally be than if we just had one hormone bind and then we had one second messenger release. No, it's more like one hormone binds and then you'll have maybe 20 second messengers that are going to be released. So it amplifies this process. And that amplification, it's going to make it larger. It increases the effect that the hormone has on the cell. And that's a way of kind of being energetically mindful. You're not making so much of these hormones and having so much of them circulate that they're not able to, they have so much of them circulate that it can kind of cause a little bit of a hormone storm, that there are too many hormones, too many messages, so that now we don't have to make nearly as many of the hormones to get the same effect by the target cell. So downregulation is when the presence of a hormone triggers a decrease in the number of hormone receptors. So if the levels of any particular hormone are really high, the cells get used to it. So they're going to become less sensitive to that particular hormone. In much the same way with upregulation, the absence of a hormone is going to trigger the increase in the number of receptors. Because the cell doesn't normally see this hormone, those target cells are going to have more of the receptors available for it, so it becomes very sensitive to it. So when the levels of a particular hormone are very low, the levels are really low, the cells are going to be much more sensitive to the hormone that's present. So the G protein is that coupler or that link between the first messenger and the second messenger. So far we've really just talked about, in the second messenger system, that the first messenger is the hormone, second messenger is something like cyclic AMP, cyclic GMP, or calcium ions, and that second messenger can be amplified even if there are just a small amount of hormones that are present in the bloodstream. Now we're kind of getting into more of the mechanics of this second messenger system. So the G protein is going to be the link between the first messenger and the second messenger. So when the hormone binds to the receptor on the plasma membrane of the cell, it's the G protein that's going to catalyze or activate the second messengers, or cyclic AMP, or the calcium ions, or the cyclic GMP. So it's activated when the hormone binds to the receptor at the membrane surface, and it's going to change the concentration of your second messenger, cyclic AMP. The more cyclic AMP we have, the more metabolic activity we'll see happening in the cell. So what we're looking at here is a little bit different than what we've been talking about. Before, we talked about extracellular receptors and the way those hormones would bind to the outside of the cell. This is looking at intracellular hormone binding. So what we have here is probably a steroid, and it could be cholesterol, it could be testosterone, or estrogen, probably most notably an estrogen or testosterone. This hormone then can bind to receptors either in the cytoplasm or into receptors in the nucleus. So it can actually make its way into the nucleus. So when it makes its way into the nucleus, it may be able to bind to hormone receptors on the DNA. When it binds to those receptors, it can either turn on or turn off genes. If it turns on a gene, then it's going to cause the DNA to make the proteins for that particular gene. So that process of transcription and translation, protein synthesis, we have that take place, and then because we have this new present protein that's available, whether it's an enzyme or a structural protein or anything else that it may be, the target cell is now going to respond very differently. So this is a little bit different process. This would have to be a lipid-soluble molecule in order to make its way past the plasma membrane. So what we've talked about, those catecholamines and those peptide hormones that have to use the second messenger system, they're not, and maybe don't have to use the second messenger system, but have to bind to the plasma membrane. They don't have the ability to go through the plasma membrane in order to directly affect the activities of the DNA, whereas this particular hormone has intracellular binding. So it will be able to, it's lipid soluble, can go through the plasma membrane, and then it can directly affect the activities of the DNA and what kinds of proteins it makes or it does not make, all because of the hormone. Other types of intracellular binding, we have some hormones, like a thyroid hormone, this is most likely a thyroid hormone, can bind to receptors not just on the cytoplasm or in the DNA, but they can bind to receptors on the mitochondria. And as you probably remember, the mitochondria is called the powerhouse of the cell because it manufactures a bulk of your energy through aerobic respiration. So when that hormone binds to the receptors on the mitochondria, it can increase the amount of energy that's produced, which can then change the target cell's response. We have more energy available, so there are more things that this target cell can do with this increased abundance of energy. Why do we have more energy? Well, we have a thyroid hormone that's binding to this receptor that ramps up the amount of energy or ATP that's made. You can have this hormone also bind to the hormone receptors on the DNA, as we saw before with the estrogen or testosterone slide, activates the genes, tells it what kind of proteins to make, and then those new proteins that are made, whether they're structural, enzymatic, whatever those proteins are going to alter the cell's activity as well. So this page and this page, these are looking at different ways of intracellular binding. And remember, not all hormones can bind intracellularly. Some of them aren't lipid soluble. The key to being able to bind intracellularly means that you have to be able to get past the plasma membrane. So you need to be at least a lipid soluble molecule. So now what we're looking at here, notice how this hormone and this hormone, they do a couple of different things with the level of cyclic AMP, that second messenger. They can't just go through this plasma membrane. They have to bind to the receptor on the plasma membrane. They can't go through it, so they can't have a direct effect on the activities on the inside of the cell. So when the hormone binds to this receptor, when we are activating adenylate cyclase, and the activation of adenylate cyclase then causes cyclic AMP to be made, The G protein is the link between the first messenger and the second messenger. So with the help of energy, or ATP, the G protein will activate adenylate cyclase. Only when adenylate cyclase is activated are we going to get an abundance of this cyclic AMP. When that happens, it can activate kinases, it can open ion channels, it can activate different enzymes. So there's a lot of metabolic change that happens in the cell as a result of cyclic AMP being produced. Cyclic AMP cannot be produced and act as a second messenger without the activation of adenylate cyclase. What I would like you to know are examples of hormones that can activate adenylate cyclase. So epinephrine and norepinephrine, when the hormone binds to a beta receptor. Calcitonin and parathyroid hormones, which both have a role in regulating calcium ion concentrations in your body, and how they respond to it. Those hormones do that. They can cause this increase. Antidiuretic hormone, follicle-stimulating hormone, luteinizing hormone, thyroid-stimulating hormone, they all respond this way, and the same thing with glucagon. Glucagon kind of does the opposite of what insulin does. If insulin decreases your blood sugar levels, then glucagon is going to increase your blood sugar levels to make sure that it stays at a level that we want it to. Now, we can also inhibit the activation of adenylate cyclase. When that happens, when we inhibit the activation of adenylate cyclase, then we're going to have more of cyclic AMP, the breakdown of cyclic AMP by phosphodiesterase is going to take place, which reduces all of the stuff that cyclic AMP would have done before. So we still have our first and our second messenger system here. We have the hormone binding to the receptor. This G protein is activated, and this time, instead of activating adenylate cyclase, we're going to activate phosphodiesterase. So when phosphodiesterase is activated, it breaks down more of the cyclic AMP. The more cyclic AMP that's broken down, the less of it that's available to activate enzymes, open ion channels, and activate kinases. So this reduction in cyclic AMP leads to a reduction in enzymatic activity. So examples of hormones that will do this are epinephrine and norepinephrine. Well, you're probably saying, wait, I thought epinephrine and norepinephrine, they both activated adenylate cyclase, which meant that ramped up the amount of cyclic AMP that was available. That's true, but look at the type of receptors. Only when they're attached to beta receptors. So if this receptor on this hormone is a beta receptor, then you will have the increase in the amount of second hormone of cyclic AMP. If, however, it's bound to an alpha-2 receptor, then you're going to have the activation of phosphodiesterase, or PDP, and you're not going to have as much cyclic AMP made. At the very beginning of this discussion, I talked about how the hormone itself is just sending a message. How that target cell receives it is dependent upon the types of receptors that are there. This is a great example of that, because if the receptor is a beta receptor, then we're going to have more cyclic AMP. If it's an alpha-2 receptor, we're going to have less cyclic AMP. So that's an important feature that I want you to kind of pay attention to. And I do want you to give me examples of hormones that can activate adenylate cyclase and hormones that can inhibit adenylate cyclase. So remember, we said calcium can also act as a second messenger. The effects of calcium levels as a result of a hormone. Once again, the hormone binds to the receptor, the G protein. Notice that for all three of these, whether it was adenylate cyclase that was being activated or it was phosphodiesterase that was being activated, the G protein still would act as a second messenger. Here, the hormone binds to the receptors, the G protein is activated, phospholipase C becomes activated, DAG, or diaglycerol, acetylglycerol becomes activated, and then you have protein kinase C activates, and because of that activation, we open up calcium ion channels to bring more calcium ions in that will act as a second messenger. In addition, wherever calcium ions are stored within the cell, that phospholipase C will cause those structures to release calcium, therefore increasing the amount of calcium available intracellularly, and this calcium that's available is acting as a second messenger, will then activate activate the enzymes needed in the cell. So it's really kind of like a very complex game of mousetrap, where you have one thing bind to the other and it causes this to happen and it causes that to happen, it allows more calcium to come in here, allows calcium to be released from here, all in an effort to increase the enzymatic activity within the target cell. Examples of hormones that do this are epinephrine and norepinephrine again, but this time they're on alpha-1 receptors, so this receptor has changed the third time. Oxytocin will do this, regulatory hormones of the hypothalamus, and you have echinocenoids that will also increase the amount of calcium, which calcium now acts as a second messenger, will increase the enzymatic activities of the cell. And whenever I say enzymatic activities of the cell or the target cell's metabolic activities, I just mean changes. Now, I'm not being very specific right now because those changes could be vast. We're talking about billions of cells inside your body, and these cells all having a different job. So that's why we're not being very specific on what those changes are. We're just saying that they're going to be changes that happen within the cell as a result of this hormone that's binding, and in this case, as a result of the increased level of calcium within the cell. So don't kind of get hung up on feeling like, well, we're not talking about what are those actual changes that happen within the cell, because there's so many of them. So we're just going to give it a very blanket statement right now and say that there are metabolic activities that take place in the cell as a result of the binding of the hormone. So G proteins and calcium ions, G proteins can, when they're working with calcium ions, they can open channels for more calcium to come in, and this is all kind of just the textual form of what we talked about on the previous slide. Release calcium ions from intracellular storage places. The G protein will activate phospholipase C, which can open up those areas where calcium is going to be stored, and also opens up those channels to allow more calcium to come in. The enzyme, the G protein, triggers this cascade of events between making more DAG and IP3 to bring more calcium inside the cell. And remember that calcium acts as a second messenger that's going to then allow for the cell to ramp up or increase its metabolic and enzymatic activities. So we have different types of reflexes. Next chapter, next set of notes that we'll talk about some of those neural reflexes. Endocrine reflexes are going to be triggered by either hormonal stimuli, which means the removing or the addition of a specific hormone, humoral stimuli, which means there's changes in the composition of extracellular fluid. Keep in mind that extracellular fluid has various different types of enzymes and various other chemicals that are in there that can alter the effects of the hormones. It can take up places on the receptor. It can do various different things to change the way that the cell responds to the hormone. And then neurostimuli is where you have neurotransmitters that come at these neuroglandular junctions, the junction between a nerve and a gland, that can also trigger an endocrine reflex. Simple endocrine reflexes are only going to involve one hormone. Complex endocrine reflexes are going to involve more than one hormone. So controls the secretion by the heart, the pancreas, parathyroid, and digestive tract. All of those are simple hormones. We'll talk about these simple hormone or simple endocrine reflexes a lot more in AMP2 when we talk about these different specific organ systems of the heart, the pancreas, thyroid gland, and the digestive tract. Complex endocrine reflexes are going to require more than one hormone, and they have kind of an intermediate step that takes place there. The hypothalamus is a great example of a complex endocrine reflex because the hypothalamus directly controls the activity of the pituitary gland. It's connected right to it and releases these releasing hormones that will tell the pituitary gland to release the hormones, and it also releases inhibitory hormones. So the hypothalamus regulates your master endocrine gland of the pituitary gland. So there are always going to be kind of a checks and balance system, and there will be two or more hormones that are released to try to be certain that the proper hormone is released at the proper time. Kind of a checks and balance or double checking, cross checking the release of hormones from the pituitary. So speaking of various different endocrine glands, this first whole half of the discussion that we had just really went over mechanisms of hormonal action. Now what we're going to do is talk about various endocrine structures and what their job is. So the first one we'll talk, and for your exam, every last one of these endocrine structures we talk about, you should know the name of the structure, you should also know, where it's found, and you should also know what it does. And most of that information I kind of have highlighted here. So your pituitary gland is also called your hypophysis, and if you'll remember from looking at your sphenoid bone in the sella turcica, you had that little indentation you can kind of stick your pinky in. It lies within that sella, so the pituitary gland just kind of hangs out in there. It has a dural sheet that locks it into position and isolates it from the cranial cavity. So although your pituitary gland hangs from the base of your brain, it's really not a part of your brain. There's kind of covering that separates it from the brain and itself as an endocrine structure. We call your pituitary gland your master endocrine gland because you have many different hormones that are released from it that will direct the activities of all of your other endocrine tissues. So it hangs off of the hypothalamus of your brain. So the hypothalamus comes at the bottom part of your brain, and we are actually going to look at that this week. We'll look at some sheep brain, I think they're in, and the way the stalk that connects the hypothalamus and the pituitary is called the infundibulum. Pituitary gland releases 9 important peptide hormones. And considering that these are peptide hormones, keep in mind that they're not going to be able to cross the plasma membrane. They're going to have to bind to membrane receptors. And by binding to those membrane receptors, a good chunk of them are going to act as a second messenger system. For many of these peptide hormones, or in fact from all these peptide hormones that are released from your pituitary gland, they're going to use cyclic AMP as the second messenger. The hypothalamus also has hormones there. You have releasing hormones and inhibiting hormones. Releasing hormones are going to stimulate the synthesis and the secretion of one or more hormones at the front part, the anterior lobe of the pituitary gland. And the inhibiting hormones that are released from the hypothalamus are going to inhibit, as the name suggests, or prevent the synthesis and secretions of hormones from the anterior lobe. Your pituitary gland, the way that we look at it and we talk about it is we talk about the front part of it and we have the back part of it. So the anterior lobe and you have the posterior lobe. Depending on what lobe we're in, the front or the back of the hormone determines, or the gland determines what hormones that we will have that can be released from there. Whether or not those hormones get released is dependent upon a part of the brain called the hypothalamus, whether it's releasing hormones or inhibiting hormones. So the rate of secretion is controlled by a negative feedback. And remember, negative feedback is going to negate the stimulus. So with positive feedback, it increases, the stimulus is going to increase the moving it towards a direction. So like childbirth, blood clotting, the presence of these chemicals calls for more chemicals to be present and until you have a clot developed. With negative feedback, like the way that we regulate body temperature, we're trying to negate the stimulus. So here is a beautiful picture of the pituitary gland. So this here would be your hypothalamus, so that's the part of the brain. This stalk here is your infundibulum. The adenohyphosis is the front part of your pituitary gland, or the anterior portion of your pituitary gland. And the neurohyphosis, we call it a neurohyphosis because the infundibulum connects directly to the hypothalamus. It's also known as the posterior pituitary gland. So things that are released from the adenohyphosis, or the anterior portion of the pituitary gland, are you have adenocorticotropic hormone, thyroid-stimulating hormone, growth hormone, prolactin, follicle-stimulating hormone, and luteinizing hormone. Make sure you know the names of all of those hormones that are released from the adenohyphosis. From the middle portion of it here, you have melanocyte-stimulating hormone. So just that little narrow band between the adeno and the neurohyphosis, you have melanocyte-stimulating hormone. And its job is to trigger the melanocytes to disperse. And then from the back part, we have antidiuretic hormones and oxytocin. Both antidiuretic and oxytocin we'll talk about at great length in ANP2. And in fact, all of these hormones, for the most part, we're going to talk about at great length in ANP2. For right now, I want you to know the name of the hormone from what part of the pituitary gland it's released and what it has an effect on. So for your adenocorticotropic hormone or ACTH hormone, it has a role on the adrenal gland, or the suprarenal gland is the same thing as the adrenal gland. It affects that, which then, so therefore it's going to affect cortisol and glucocorticoids. Thyroid stimulating hormone, that's a pretty easy one. The organ it affects is right there in the name of the hormone. It's going to affect your thyroid gland to produce different hormones, primarily T3 and T4, and we'll talk about that more in this discussion. Growth hormone has an effect on the liver and the development of bones, muscles, and other tissue that's needed for development. Prolactin has an effect on your mammary glands. Follicle stimulating and luteinizing hormone has a role and effect on the testes, which will cause them to produce inhibin and testosterone if you have these structures, and your ovaries for the luteinizing hormone and follicle stimulating hormone, which have a role in the production of estrogen, progesterone, and inhibin. For oxytocin, both in males and females, it can cause, for females, causes uterine smooth muscle contractions and mammary glands as well. For those contractions, it has an effect on those. For the males, smooth muscle in the ductus deferens or the vas deferens and the prostate gland. And then antidiuretic hormone directly affects the kidneys. So know this slide well. There's a very good chance that you will have a short answer question that will ask you to list the 9 hormones released from the pituitary gland and what is the effect, what organ system or tissue do they affect, and what is the job that they do. So the neurohypophysis contains unmyelinated axons of the hypothalamic neurons. Remember I said it kind of connects directly to the hypothalamus there. And the 2 hormones that are really released there are the antidiuretic hormone that affects your kidneys and your oxytocin, or is kind of known as your love hormone, and it primarily affects uterus and prostate and vas deferens relaxation and contractions. So moving down a little bit further south, so we started up at the pituitary hormone, the master hormone, and now we're moving down to the thyroid hormone. Your thyroid hormone lies anterior to your thyroid cartilage of your larynx, so right in your cervical region, if you will, if you remember your anatomical objectives, right in your throat region. It has 2 lobes, and it's connected by a little isthmus. There are thyroid follicles that have cuboidal epithelium, so they're shaped like little cubes, short, squatty little cells. They are connected by capillaries that will deliver nutrients and hormones, and those capillaries can also secrete waste products and metabolic waste. Thyroid stimulating hormone is produced from we talked about from the pituitary gland, and when thyroid stimulating hormone is released, it can cause the thyroid, absence causes the thyroid follicles to be inactive. So if there is no thyroid stimulating hormone that's released, then those follicles are not active, and they can neither make or secrete any of those T3 or T4 hormones. Thyroid stimulating hormones binds to receptors, membrane receptors that can either be on the plasma membrane, but most notably those thyroid hormones can go through the plasma membrane and bind to the mitochondria, or they can bind to the DNA itself. When that happens, it activates key enzymes in thyroid hormone production, notably T3 and T4. Thyroid hormones affect most of the cells in your body. As I said before, those thyroid cells can bind to receptors in the cytoplasm, in the mitochondria, in the nucleus, all places within the cell because they are lipid soluble. In children, thyroid hormones are essential to their development for skeletal, muscular, and nervous system development. Thyroid hormones, because they bind to the mitochondria and they directly have an effect on how much energy or ATP is produced, have what we call a calorogenic effect. The cells consume more energy, resulting in increasing in heat generation. This calorogenic effect is also responsible for strong, immediate, short-lived increases of the rate of metabolism. So you've probably heard of hyperthyroidism and hypothyroidism. So those individuals that don't make enough of these thyroid hormones were considered hypothyroidism. They had difficult time losing weight or maintaining healthy weight balances. Energy levels are usually pretty low. For hyperthyroidism, it's kind of the opposite end of the spectrum. They have a difficult time gaining weight because they're always losing. They have a lot of energy that's made, but it's not made in a useful sense. So sometimes they also deal with being tired, but their weight is also much lower. There are different types of drugs that can be taken to either balance out for either hypo or hyperthyroidism. C cells or clear cells of the thyroid gland are going to produce calcitonin, and calcitonin along with parathyroid hormone help to regulate calcium concentrations in the body. Parathyroid hormone responds to low calcium concentration. So the two hormones that are responsible from, and the parathyroid hormone is released from the parathyroid gland, which is on the backside of the thyroid gland. It's kind of got its own little section back there. Calcitonin, parathyroid hormones are both responsible for regulating calcium levels. The way that parathyroid hormone works is that if the blood calcium level is too low, parathyroid hormone will increase the activities of the osteoclast. And if you remember back from the skeletal chapter, the osteoclast will break down the osteocytes and release or liberate calcium into the bloodstream. So parathyroid hormone's job is to release calcium, and it only is secreted by these chief cells, or these cells in the posterior surface of the thyroid gland, if the calcium concentrations are too low. So with that being said, if parathyroid hormone is responsible for liberating calcium in the skeletal system, what do you think is the job of calcitonin? It would be the opposite job, right? So calcitonin's job is to take the abundance of calcium that's available in the circulatory system and put it into the skeletal system to be stored. So the effects of parathyroid hormone stimulates osteoclast activity because we want to put more calcium into the bloodstream, enhances the reabsorption of calcium at the kidneys so we can retain more calcium, and when it does that, it reduces the amount of urine that's lost. So we want to retain this calcium to increase our blood calcium levels. And I don't even need to discuss why calcium is so important. We've already talked about how in almost every organ system that we discussed this semester that we've seen the role of calcium. So you probably have a pretty good understanding of why we need to maintain blood calcium levels in the same way that blood sugar levels should be maintained. It also stimulates the formation and secretion of calcitorol at the kidneys, which enhances calcium and phosphate absorption from the digestive tract to make sure that we have enough calcium that's available to bring our blood calcium levels up to where they need to be. So big take-home message here is that there are 3 ways that parathyroid hormone can increase the amount of calcium that's available. And the job of parathyroid hormone is to ensure that it's released when blood calcium levels are too low. Calcitonin, on the other hand, job is to put excess calcium ions into the skeletal system to be stored, and then they can later on be released with parathyroid. So now going a little bit further south to the adrenal glands that sit like little hats on top of the kidneys. They're divided into the superficial or suprarenal cortex. So your book and your notes are going to say suprarenal or adrenal. Supra means on top of, renal, kidneys, so they sit on top of the kidneys. The superficial aspect of it, or the outside of it, stores lipids, especially cholesterol, fatty acids. It makes steroid hormones, adrenocorticosteroids and corticosteroids. The inside portion of your adrenal gland, it secretes activities that are controlled by the sympathetic division of your autonomic nervous system. And remember, you had your sympathetic and your parasympathetic divisions of your autonomic nervous system, your fight or flight and your rest and repose. The hormones that are released there or produced are epinephrine, also known as adrenaline, and norepinephrine. And when these hormones are released, the changes that happen are going to persist for several minutes. So they're not going to be long-lasting hormones like we saw for estrogen or testosterone or for thyroid hormone. These are going to be hormones that are not bound to transport protein. So they are free hormones, they circulate, they'll allow the cells to respond very quickly, but after several minutes, they're going to be broken down and recycled. So let's look at those different zones of the suprarenal glands. So the zona glomerulosa is the outer region of the suprarenal cortex. It produces mineralocorticoids. Most important one we're going to talk about is aldosterone. Aldosterone stimulates the conversion, the conservation of sodium ions and the elimination of potassium ions. When you think of the adrenal glands, a lot of what they primarily do, you probably already think of adrenal glands, you think about adrenaline, epinephrine, and norepinephrine, but it also has a role, and remember, they sit right there on top of the kidney, so they should help the kidneys kind of do their job. They also have a role with salt and sugar releasing and maintaining the salt and sugar balance. So aldosterone, it's when that hormone is released from this outer region of the suprarenal or the adrenal gland cortex, the outside portion of it. When it's released, it's going to tell the body, hey, we need to retain more of these sodium ions, so that when the sodium ions and the potassium ions are going right down below to be filtered out, some of which by the kidneys, it keeps those kidneys from, it closes some of those channels. And this will all make more sense when we get to AMP2, but it closes some of those channels so you don't lose as many of those sodium ions, and you can retain them for various aspects or requirements that are needed in the body. What it will also do is it will increase the sensitivity of salt receptors on your taste buds, so you'll be more sensitive to salt, and your body is going to want to retain more of these sodium ions. Aldosterone secretion responds to a drop or a decrease in sodium ion concentrations, or a drop in your blood volume, or a drop in your blood pressure. A lot of times what happens here is that when you have a drop in your blood volume, the volume of blood that you have, what's going to also do is it's going to drop your blood pressure, because you don't have the amount of blood that's hitting the walls of those vessels and hitting those barrier receptors to give the blood pressure what it needs to be. Salt is going to, or water follows salt. So if you're releasing a lot of salt, chances are you're also releasing a lot of water. The water is going to follow that salt. And that's all as a result of, you know, what we talked about with hypertonic and hypotonic solutions. So if you're losing a lot of salt out, then chances are water follows salt. You're also losing a lot of water. If you're losing a lot of water, one of the things that's going to be affected primarily is your blood pressure and your blood volume. If your blood volume will go down, your blood pressure will go down as well. So that's why these things are related to one another. Sodium ion concentration, blood volume, and blood pressure. So if we have a drop in any one of these, that will stimulate your body to retain those sodium ions, and by retaining those sodium ions, remember, water is going to follow it, will increase the amount of water into the body, thereby increasing the amount of water available in the plasma, therefore bringing your blood pressure back up to where it needs to be, because you brought your blood volume back up as well. And it can also, because we eliminate potassium ions with aldosterone, if there's a rise in potassium ion concentrations, then you can also have this hormone that's released. Zona fasciculata produces glucocorticoids, or the sugar part of this. For example, cortisol, hydrocortisone, and corticosteroid, their whole job here is they kind of give out an inflammatory response. Their secretion is regulated by negative feedback. Remember we talked about glucocorticoids being the sugar part of this. It accelerates glucose synthesis and glycogen formation, so that the more you store more glycogen available. Any glucose that's available, you can store it as glycogen, and you can also make more glucose when these glucocorticoids are released. It has anti-inflammatory effects, so it inhibits white blood cells and other components of the immune system, so it kind of helps to regulate the immune system with the anti-inflammatory effects. The zona reticularis is a band between bordering the supraadrenal glands. This is where we produce our androgens. So really the adrenal gland, I've heard people refer to them that they have a job in regulating sex, sugar, and salt. The androgens can be produced when adrenocorticotropic hormone is released from the pituitary gland. And androgens are things like testosterone is a good example of an androgen. Now to the middle part of our adrenal gland, we have two types of cells that can be released there, and they're both of them epinephrine and norepinephrine. One is adrenaline, and the other is noradrenaline. And now we're actually going to go back a little bit further north, and we're going to go back up to the brain, to the pineal gland. The pineal gland, and we'll look at this gland today and this week in lab class. Pineal gland, it's like a little cherry pit that just kind of hangs out in the roof of the third ventricle of the brain itself. It contains pinealocytes, and the job of these pinealocytes is to make this hormone melatonin. Melatonin is responsible for setting your circadian rhythms here, so your day-night cycles. Melatonin can also inhibit reproductive functions in some animals. Some animals, like birds, they have, some species of birds will have a very small hole in the front part of their cranium that will allow for light to go directly to their pineal gland. So even the smallest amount of sunlight that's available can go to their pineal gland and then trigger their circadian rhythm. So you've probably noticed that the birds in your neighborhood around the springtime and the summer, they're up long before the sun is. At least the birds in my neighborhood, in my yard, are up long before the sun is, because any small amount of daylight triggers their circadian rhythm, and they're perceiving daylight. You'll also notice that a lot of animals are only reproductive on certain times of the years, a certain time of the season. They have their reproductive season, and their little babies are born in the spring or whatever. Humans, we don't really rely on our pineal gland for that reproductive function as much anymore. All right, so we're going to stop here, and then we will finish up with the pancreas and talking about the rest of the endocrine glands, and then we will also be able to start our last section of notes this week as well. All right, questions, comments, concerns? I will see you all this week, and no quiz this week. No quiz. I might give you an online something or another quiz, but no quiz in class. All right. Have a great day.